All right, hi guys. I am not going to show my face today because I look really bad. And yeah. So anyway, hope you guys are doing well. Hopefully you guys have turned in the project of the genetic disorder project. We're going to move on and we're going to talk about mutations in chromosome abnormalities and genetic engineering. That's our last unit in genetics. So there's a lot of cool things we can do. We can research in here. When you guys see or hear the word mutation, you think of something like crazy like this. And yes, this is a mutation, but there are some mutations that are good, and we're going to talk about those as well. The other thing is you can either take notes from my follow along sheets, or you can do your own notes. I know that some people like to do their own notes. Some people are really, really good at doing their own notes. So if you feel like you do really well with making your own notes, you can do that instead of doing the follow along sheet. Whatever you guys want to do, whatever you think will help you more, but you do have to turn them in. All right, so with that being said, we're going to move on. So for most genes, there are at least 2 alleles in the gene pool, one from the mom and one from the dad. Sometimes there's more than 2, like multiple alleles. In rabbits, there's a lot of different alleles for fur colors, but they only get 2. Alleles that are not passed on to offspring can be lost. New alleles result from mutations. So if we did not have mutations, we wouldn't have genetic differences. A mutation is a permanent change in genetic material that is passed from parent to offspring. And like I said, not all mutations are bad. They're a source of genetic variety. There's 2 types of mutations we're going to talk about. One is a gene mutation, and the other one's a chromosomal mutation. So one's more microscopic, like you're looking at specific codons, like those sets of 3 on RNA, where chromosomal, or you're looking at a whole section or chunk of DNA. So a mutation is a change in a DNA sequence. Mutations are the source of different alleles found in species or populations. Mutations can occur in any cell of the body, but if they end up in gametes, the mutation can be passed on to offspring. There's mutations that go on in our bodies in everyday life. I think it's 1 out of 100 billion cells that actually will mutate, and usually if the body catches them, they'll kill them off, or they do self-destruction. But if it ends up in gametes or sex cells, that is when they're passed down. And that's when you can either have changes in a population, say you had a mutation for different fur color, and the fur color helped them blend in more, that's a good mutation. So not all mutations are bad. Not all you're going to end up with like 3 heads. A codon is a set of 3 nucleotide bases that specify an amino acid. So if you guys remember when we were looking at DNA, you have your that represents AUG and RNA, and then RNA codes for methylene. DNA, if you have the codon GGT, that would be CCA. That is the codon for proline. All these are amino acids. So there's silent mutations in DNA that actually don't change the resulting protein. I don't know if you guys remember that genetic code. It was like that circle where you had to like work out and read, or, we did the codon bingo. This would be a silent mutation. So CAA, the RNA would be GUU, that codes for valine. CAT, in RNA, that represents GUA, that also codes for valine. So that's a silent mutation. It doesn't actually change the protein. So you can have mutations and not even know it. A missense mutation causes one amino acid in the protein to change. So this one, a missense, if there is an insertion, or there's an A instead of a C, everything else is the same, it changes one amino acid. So here's the original strand. This is the original strand of DNA, and these are the amino acids that they code for. On this strand, instead of A, you have a C, So instead of coding for glutamine, it codes for aspartic acid. So that would be a missense. It doesn't make sense. And it might not fold properly. So remember, when these amino acids kind of are being built, they kind of fold on top of each other. They make a certain shape. So this one might fold wrong. A nonsense mutation inserts a stop codon and shortens the resulting codon. So here's your normal DNA sequence. Here would be the normal RNA that would be linked to it. And the normal amino acids coded for that are these like glutamine, glutamine, glutamine. But if you have a missense, say instead of the C, a T was inserted, and ATC, the RNA is UGA, and that actually is a stop codon. So it just puts one amino acid there and stops. So the protein's not even made. So some people are missing proteins out of their body. It's probably a mutation where the stop codon was put in place and then their bodies can't create that protein. Like lactose intolerant people do not have lactase. That's an enzyme or a protein that breaks down lactose milk. And so when they don't have that, their stomach gets upset because it won't break it down. That might be because of a nonsense mutation. And then a frame shift is where either you have a base added or it's removed. So mutation changes how bases are grouped into codons on the reading frame of the gene. So remember, if you have DNA and they're red in threes, this is like the normal amino acids that would be made from this strand of DNA. But say you had, instead of, I'm seeing where it was inserted, a frame shift, one of the DNA sequences results in a frame shift. So even in the beginning, there's one added and it shifts the frame down this way. So there's like none of these are the same. It just is a frame shift. It completely messes up the whole reading frame. And I did this like in as example. Say you had, you know, these were codons. Say that these, it's a sentence, but the fat cat ate the rat. Say those are codons. And say instead of a T nucleotide, an A was put in place. And so it still would read the, and so that amino acid would be the same, but FAA would not be the same. That would, so it doesn't make sense anymore. It's, and even this one, you have a leftover one, because remember, they're red in threes. So the whole reading shift goes down that way, like cat, CAT, the T actually will go to TAT, 8 would be ATE, and it's, you know, it doesn't make sense. Or that happens if you remove a nucleotide. And so, you know, it's had the fat cat, say you remove the A, everything would shift the other way. So those are frame shifts. So they really mess up the whole amino acid base, and it can make a totally different protein. Chromosome mutations are on a bigger scale. So we talked about gene mutations, which are in specific nucleotide bases. Now we're talking about chromosomes that it's basically a gene. Like, you know how you have DNA. So we were looking at like the little portions of DNA. Now we're looking at whole sections of DNA that can be altered or can totally mess up the entire chromosome. So these are chromosome mutations. Here's the different types. So this is the normal chromosome. And, you know, it goes A, B, C, D, E, F. A deletion would be part of that chromosome is missing. So B is not there anymore. So that's a deletion. Here's the normal chromosome. And then a duplication would be part of the chromosome just gets duplicated for some reason. It adds an extra section of that. An inversion, if you have the A, B, C, D, E, F, it kind of pretzels on itself. And then the B and C are backwards and they're pretzeled on the other way, and the D and E are backwards and it's on the other side of the thing. So that's an inversion. And then a translocation, here's your original chromosome, A, B, C, D, E, F, and a completely different chromosome comes in and it gets stuck together. And so part of one goes on to the other. That's called translocation. So all these are pretty bad types of you know, DNA. So deletion removes a segment. Duplication causes genes to be repeated. Inversion flips the order of genes on the chromosome. And a translocation moves segments from one chromosome to another. So changes in chromosome numbers are also an issue. Remember when we talked about meiosis, making sex cells, and makes gametes. Chromosomes separate during meiosis or mitosis. Chromosome abnormalities occur when chromosomes fail to separate. That's called non-disjunction. And it usually happens, if it happens, usually the embryos are miscarried, but an exception is trisomy 21 or Down syndrome. So basically, if you look here, instead of going separate ways, these don't separate and they end up on another gamete. So remember, it's supposed to be half and half. And you know, this side has 1, 2, 3, 4, 5, 6, 7, 8 chromatids. This side only has 4, where it should have 6. So 1, 2, 3, 4, 5, 6 chromatids on that side and 6 on that side. Chromosomes together, you have 3 and 3. This side, you only have 2, so you're missing a chromosome, and this one, you have an extra. This is another example of non-disjunction. And then genetic engineering, this is really, actually, I'm going to do that later. So I'm going to stop there. If you guys want to get, so just stop there, and then tomorrow we'll finish this up and do a worksheet. So you don't have to turn anything in today, just stop there. Or if you want to keep going, look for the part 2 tomorrow. All right. So I don't want to like keep going because I know it gets boring after a while. All right. Bye.